Hi guys, hello. Today's our topic is AIDS. Let us discuss about AIDS. Before entering into the today's discussion about AIDS, let us discuss about the magic of nucleic acids in central dogma. Central dogma describes how the flow of genetic information occurs within a cell. DNA flows into mRNA, mRNA flows into proteins. The flow of genetic information from DNA to RNA is called as transcription. The flow of genetic information from mRNA to proteins is called as translation. In case of AIDS, there is a process exceptional to the central dogma. Reverse transcription occurs. It involves the conversion of RNA back into DNA, which is not a part of typical flow of genetic information DNA to RNA, RNA to proteins. Reverse transcription occurs in certain viruses like HIV, where genetic information stored as in the form of RNA. The enzyme reverse transcriptase converts their RNA genome into DNA, which can integrate into the host's genome. Let us know the difference between HIV and AIDS. HIV is a virus that attacks on our immune system. AIDS is a group of symptoms of severe damage of our immune system. AIDS is a acquired immunodeficiency syndrome, deficiency of immune system that acquired during the lifetime. HIV attacks the body's immune system, making it difficult to fight against infections and diseases. Last 25 years, 25 millions of people were killed by the HIV. HIV is a chronic disease in the sense it is a condition of long-term health issue. It is a non-congenital disease, means this disease is not inherited from parents at birth. HIV is a viral disease belongs to the class retroviruses. HIV virus having two identical molecules of RNA and with one reverse transcriptase enzyme. HIV was first reported in the year 1981. How AIDS transmission occurs? By sexual contact. Unprotected sex leads to AIDS. By transmission of contaminated blood and blood products. Sharing of needles, syringes with contaminated blood. Mother to child transmission during pregnancy, infected mother to placenta during childbirth. During breastfeeding also, AIDS transmission occurs. HIV can be transmitted through organ transplantation. HIV positive donor to recipient. In today's session, now let us discuss about symptoms of AIDS. Fever, diarrhea, lymph nodes are swollen. Due to the decrease in T helper cells, person starts suffer from infections like mycobacterium, virus, fungi, parasites like toxoplasm. Weight loss. These all are symptoms of HIV. Now our cinema is about HIV life cycle. Understanding HIV life cycle and several stages of HIV is crucial for developing the effective treatments and preventive strategies. CD4 cells is a glycoprotein present on the surface of T helper cells, macrophages and monocytes. Macrophages are called as a HIV factory. In the given diagram, animal cell is a macrophage cell. Suppose imagine. First step of HIV life cycle is attachment. Attachment of HIV virus to the host cell. CD4 cells. CD4 cells present on the macrophages. CD4 cells are the receptors for HIV virus. Now HIV virus come and binds to the CD4 cells. And next step is fusion. HIV fuses with the host cells cell membrane and releases its genetic material RNA into the host cell. Reverse transcription. Third step is reverse transcription. The RNA is converted into the DNA by the enzyme reverse transcriptase, which is the process opposite to the central dogma. Fourth step is integration. Viral DNA incorporates into the host genome with the help of integrase enzyme. Next step is transcription. The integrated DNA is transcribed into new mRNA molecules. Next step is translation. RNA molecules are translated into the new viral proteins. Assembly. Next step is assembly. New virus particles assembled and using newly synthesized proteins and RNA molecules, development of new virus particles takes place. This is a assemble. Next step is budding. The new viral particles bud out of the host cell and releasing infection virus. These viruses are very happy. Their virus generation now it's increasing. That's why HIV virus are feeling very happy. Okay, now let us see 3D animation of HIV life cycle.
When HIV first enters the human bloodstream, the virus circulates throughout the body. The protective surface of HIV is studded with particular proteins called GP120, and inside is a capsid that contains the viral RNA and viral enzymes. Initially, the GP120 protein on the virus is only able to bind to a cell surface protein on the macrophage called the CD4 receptor. A second receptor protein, CCR5, must also be present on the macrophage for the virus to enter the cell by endocytosis. Viral RNA and enzymes are released into the cell's cytoplasm. Reverse transcriptase uses the viral RNA to synthesize first a strand of viral DNA and then the complementary DNA strand. The viral DNA then moves to the nucleus of the cell, where it integrates into the host cell's DNA. Transcription of the DNA within the nucleus now results in the production of viral RNA. This viral RNA can serve as the genome for new viruses and can be used as messenger RNA to produce viral proteins during translation by the ribosomes. Complete viruses are assembled and released from the cell via exocytosis, which causes little harm to the macrophage. HIV cycles through macrophages over a period of years and continues to multiply while doing little apparent damage to the body. Eventually, the gene that codes for the GP120 protein is altered by mutation. The altered GP120 protein changes its co-receptor allegiance and now binds to a different co-receptor, CXCR4, which is found on the surface of CD4 plus T cells. The same processes occur within the T cells, resulting in new virus particles. However, as these new viruses leave the T cell, they rupture the plasma membrane, which kills the cell. As these newly released viruses invade and destroy other T cells, the body's immune response is weakened, and this leads directly to the onset of AIDS.
Diagnostic tests for HIV. First test is ELISA, enzyme linked immunosorbent assay. ELISA test is based on antibody antigen mediated mechanism. ELISA test detects HIV antibodies present in blood. In presence of antigens, in response, antibodies are produced. Second test is Western blotting test. Test for proteins. Western blotting test confirms HIV positive by detecting specific antibodies. Rapid test. It provides quick results using oral fluid or blood. PCR test. Polymerase chain reaction. PCR detects HIV genetic material is RNA or DNA. Viral load test. Viral load test measures HIV RNA levels present in blood. CD4 cell count. CD4 cell count helps to measure immune system health. These all are the tests help to diagnose HIV infection, monitor disease progression, and estimate effective treatments. Prevention of AIDS. Friends, AIDS has no permanent cure, but it can effectively manage it through ART, antiretroviral therapy. ART, antiretroviral therapy suppresses viral loads and prevent progression to AIDS. Prevention is better than cure. HIV spreads due to conscious behavior. It is not something that occurs inadvertently like pneumonia, typhoid. Safe transmission of blood products, strict screening of blood donation. In India, National AIDS Control Organization (NACO) and other non-governmental organizations (NGOs) are doing a lot to educate and bring awareness about AIDS. Safe sexual practice. Free, free distribution of condoms. Use disposable needles and syringes. Avoid sharing used needles and syringes. Friends, if you are confused or puzzled with any topic in 10th science and 12th class bio, do comment on comment session. Bye bye. Thank you.